thank you everyone for being here today. For those of you who don't know me yet, I'm Alex Lemens, the director of our Molecular and Genomics Informatics core facility. We also have on the line the entire team from Lumicks, who's going to be presenting about their technology and kind of how you can use it for different molecular interactions. But just the general housekeeping items before I turn it over. If you have any questions, please put them into the chat or the Q&A box. The team will be trying to answer what we can live, and anything else we'll answer at the end. And with that, I'll turn it over to the Lumicks team and hear about their awesome technology.
Thank you, Alex, for inviting us, and hello, everyone. So today, this talk is divided into two parts: the DSM part and the cell ability part. I will be presenting the DSM part, and the title of our talk today is "Exploring Molecular and Cellular Interaction Through Light and Sound: A Critical New Tool to Understand the Mechanism and Modes of Action from Single Molecule Dynamics to Cell Avidity." So my name is Samrat Dara, and I am an application scientist at Lumicks. So without further ado, I'll start my presentation. So today I'm going to be introducing you to the C-trap technology and its application. But before that, I will briefly touch upon why we want to study biology at the single molecule level. And at the end of my presentation today, I will mention to you how Lumicks can support single molecule research in your lab. So why single molecule? Because biological interactions are complex and heterogeneous in nature, where the biomolecules undergo structural and conformational changes, as you can see in the video here, which is a cartoon video representation of DNA replication. We see the DNA and protein molecules undergo conformational changes to perform a particular biological function, which is replication here. So to exactly understand biology at the fundamental level, it is extremely important for us to study how these molecules changes their conformation and structure and evolve over time. So a lot of data in the last multiple decade has already been provided. The crystal structure data gives us angstrom scale resolution of the biomolecules, but however, the data does not tell us about the dynamics of this like how the structure evolve over time. Simultaneously, we know a lot about the biological interaction using bulk functional assays. They tell us about the dynamics of biological interaction. However, these assays are performed over millions of molecules. So what we see is basically the start and the stop of a reaction. It doesn't tell us the entire mechanism or the pathway going from the start to the stop, which is extremely essential to understand the mechanism of how biology works. And it is why it is extremely important, because uh, there are transient states which are involved in a biological interaction, which can get averaged out in these ensemble assays, which may involve piconewton to nanometer scale interactions. And that is what I'm going to talk about today. So we asked a question to ourselves that what if we have an instrument using which we can visualize a biomolecule? For example, here the animation on the left hand side shows a cartoon protein molecule is walking over the DNA. And what if you can visualize them? Because seeing is believing. Also, what if the system has the property or the facility to assemble multiple biomolecules in the assay? Because a biological interaction can have more than one molecule there. And finally, since biological interaction, as I mentioned, undergoes conformational and structure changes, what if we have the facility in the system using which we can manipulate and measure interaction over the biomolecule? As you can see here, this is a fork junction where this complex protein binds very slowly and moves along the DNA. But when the mechanical force is applied on this DNA, this protein molecule starts to move a little bit faster. So this is basically, in essence, this is the essence of the C-trap technology. And I want to quickly go over this. Like the C-trap integrates an optical tweezer with optical microscopy, and the optical tweezer in the C-trap is being inspired by Arthur Askin, who invented and got the Nobel Prize in optical tweezer in 2018. So an optical tweezer is an instrument where focused beam of light is passed through an objective through a transparent bead or a particle. So once the light passes through this transparent beam or a particle, it undergoes change in the momentum because of the change in the refractive index. And this change in the momentum pulls the trapped bead towards the center of the focused laser. And this allows us probing biological system at the piconewton and the nanometer range. So essentially, C-trap, this is how a C-trap looks like. So a C-trap has four different major components. It has the microscopy unit, which is used for visualization purposes, and it has the optical tweezing unit, which is for manipulation and measurement purposes. It has a microfluidic unit, which is used to assemble multiple biomolecules in the assay. And finally, all these three units are being interfaced or operated using a software called Blue Lake. Blue Lake software is written in Python, so it is an open source. So a user can have access to the code, which allows them and give them the flexibility not only to write their own experimental scripts, but also they can modify the existing scripts. I'm sure this feature in the C-Trap will be appreciated by a lot of home build people who build their own optical tweezer. And as we know that it's a kind of enormous amount of work writing different, making the entire system work robustly when you are doing it like if it is a home built system. So let's talk about the micro. At first, talk about the microscopy unit in the C-trap. The microscopy unit in the C-trap can be divided into two different parts depending upon the application. So type 1, the optical tweezer in the C-trap can be combined with a confocal, and this design is suggested for assays which are more fluidic in nature, or in three dimension. For example, if somebody is interested in doing protein DNA interaction experiment, then we suggest having a C-trap where the optical tweezer is combined with a confocal microscope. The confocal microscope can have up to 3 colors with a single photon sensitivity. And also, this confocal can be later on upgraded to a STED system for super resolution microscopy. The type 2, when the application is related to more surface assays. For example, if someone is interested in doing studying cell mechanics or protein or interaction of kinesin with a microtubule, we suggest this setup of the C-trap where the optical tweezer is integrated with microscopy unit, which has the wide field, turf, and interference reflection microscopy. I will show a data on interference reflection microscopy later. It's a label free technique. The second important unit in the C-trap is the microfluidic unit. As you can see, we call this unit as a U-flux. It has a pressure box, and using this pressure box, the pressure can be applied across the microfluidic channel. Here, this shows the microfluidic channel, which has 5 channels, and upon applying the pressure, the the buffer or sample through these different channels flows under they have a laminar flow. So this flow cell is construction of this flow cell is based on the laminar flow, which means there is no physical barrier between this channel, and a user can move biomolecules or particles from one channel to the other depending upon the application. So I will quickly show a video which will give an idea about how the microfluidic flow cell works. So I'll quickly. So this is a real-time experiment which was done using fruit colors. So as we can see, 5 colors here and 5 channels. And as we can see, if we close one channel, the the other color occupies the volume of the other channels, which further proves that there is no physical barrier within the channel. Moving on, as I said, the C-trap also has a optical tweezing unit which is used for manipulation and measurement purposes. So a C-trap generally comes under two configuration. Either you can have a dual trap where there are two traps, or a quadruple trap where there are four traps, which essentially depends upon the application. And the video here shows that how simple it is to trap a bead. So these are the beads, and this is where the optical trap is focused, and you can see within second the bead is trapped within the optical focus. So before going into the application, I will, as I have mentioned, that the user has the option and the flexibility to choose uh, dual trap or a quadruple trap. They can also choose a single trap, which depends upon the application. For example, assays where only one handle is required. For example, when we want to study the interaction of kinesin with microtubule, we only need a single trap. So in that case, a mono or a single trap can be used. Similarly, for assays where we want to probe the cells sitting on the surface, in that case also we can use a single trap. Dual trap is essentially used for assays which needs two handles. For example, when we need two handles to tether a DNA molecule here. For essentially, like when we want to tether a filament between two beads and study interaction of biomolecule with that filament. For example, protein DNA interaction. User also has the flexibility to get a quadruple trap where there are four traps or four handle. Essentially, this design is used for applications which are uh, like where the interaction between the filament is necessary. For example, how the protein interact across the holiday junction. So to better understand how a C-trap works, I will go through an animation video which shows the workflow of a C-trap. So this is how a C-trap look like. This is the U-flux where the pressure is applied through the 5 channels in the microfluidic through the flow cell. As you can see here, there are 5 channels, and these are the schematic representation or animated representation of all the 5 channels. On top is the bead channel, and the bottom is the buffer channel. So the first action is to focus the optical trap in the bead channel and get 2 bead trapped into the optical trap. This bead could be strep-tavid encoded, and then the optical trap is moved into the DNA channel where the bioattenuated DNA could be tethered in between the two traps, and then the uh, optical trap is stretched, and we get a worm-like chain model which verifies that there is a single DNA molecule between the trap. After we know that there is a single DNA molecule between the trap can be moved into different protein channels so that the protein can bind to the DNA molecule. And as you can see here, this protein schematically they are color coded, which means that they can give different fluorescence signal. And once the protein is assembled on the DNA molecule, the trap can be moved into the buffer channel for confocal scanning. Here, the confocal scanning is done across the length of the DNA, and while it scans the DNA, different fluorescence signal is generated from this protein. So essentially, what we can find out from this experiment is first the interaction forces of this protein molecule with the DNA, and we can also determine the stoichiometry of this protein molecule on the DNA, whether it's a 1 mer or it's a 2 mer, and that's basically that information usually we get by the photo bleaching signal. So one thing I wanted to just mention that the entire workflow which I have shown before, it takes about 5 to 10 minutes to perform the entire assay. So just wanted to mention here that like C-trap is extremely easy to use to perform a single molecule experiment. So over the past 4 or 5 years, the C-trap has been used to address multiple questions in biology, not only limited to these. And I'm going to talk about few of these applications today in this talk. So I will start with protein DNA interaction. So C-trap is extensively used to study protein DNA interaction to address multiple questions from DNA repair to DNA translation. And as I can say, it can also be used to study holiday junction, where four different traps are used to study how a protein interacts across the holiday junction. So I will quickly go over how a protein DNA interaction is done with real data to show you how it is done in C-trap. So essentially what's done is, at first, in this case, a streptavidin coated bead is, two streptavidin coated beads are trapped in the dual trap. And once they are trapped, the protein of interest which you want to study is flown through the DNA. And as we can see here, the video I will quickly show you is an experiment where, as we can see, these are the two beads which uh, tethers a DNA molecule. Since the DNA molecule is not labeled fluorescently, we do not see the DNA molecule, and we flow the protein molecule through it. And as we can see, the protein binds all across the DNA. And this type of assays can be used to address multiple questions from DNA repair, DNA organization, DNA translation. If if someone is interested in studying how a damaged DNA due to interaction with a cancer drug reacts to a particular type of protein, those type of assays can be done pretty easily using a CTRA. So what we so far I shown the video, and we did see the interaction of the protein with the DNA. But what let's talk about what quantitative information which we can find out from these type of assays. So from the previous, so on the left hand side, this is a snapshot of the chymograph which we obtained from the previous experiment. So let me briefly talk about what a chymograph is. In a chymograph, the x-axis represents the time and y-axis represents the fluorescence signal along the length of the DNA. So if a protein molecule or a fluorescent protein molecule binds to exactly one location on the DNA and does not move from there, then we see a line, a, like a horizontal line parallel to the time axis. But if the protein binds to multiple locations on the DNA, or there are multiple binding sites for the protein, then we can see a smudge. We can see that the fluorescence signal is all across vertically, which means the protein has bound to multiple locations. Another very interesting thing which we can measure from this chymograph is the location where the protein binds on along the DNA with the accuracy of approximately 15 base pair. And this is pretty interesting because we get to know where the protein binds without going through um, like approaches like DNA sequencing and all. Another important thing that can be measured from this assay or chymograph is the kinetic off rate and the kinetic on rate. Kinetic on rate is where we can directly measure from the time, and the kinetic off rate also we can measure just dividing by the duration time it binds to the particular DNA molecule. As I mentioned, that we can also measure the stoichiometry of the protein molecule which is bound to the DNA just by studying the photobleaching. As you can see here, if here there are two photobleaching states which corresponds that there are two mers or two protein molecules are bound to the DNA. So as a case study, I will present to you a data where off-target binding of Cas9 or CRISPR-Cas9 on the DNA was studied. I think all of us know that CRISPR-Cas9 it's a very popular area of research nowadays because of its application in gene editing, and it could have very important implications, and we want to make sure that the gene editing is very specific. So this group from Imperial College London, what they found out is that when the CRISPR-Cas9 complex it binds to the DNA, when the DNA molecule which is tethered between these two beads are kept at a very steady or a minimum force, I would say, and it binds to exactly one location. And they wanted to study that what happens if the DNA comes under tension, because uh, in our day-to-day everyday life, DNA is undergoing multiple conformational changes, and and structural changes. Due to that, there is some tension on the DNA. So that was mimicked by them by applying force on the DNA molecule by stretching these two beads, which are streptavidin coated, and the DNA was a lambda DNA, which was biotinylated. And what they found out is that when the DNA was stretched, the CRISPR-Cas9 binds to multiple locations on the DNA, or there are a lot of off-target binding of CRISPR-Cas9 on onto the DNA molecule. This is a more quantitative representation of what they saw. Again, this is a chymograph. The y-axis represents the fluorescence signal along the length of the DNA, and the x-axis represents the time, where different type of forces or various forces were applied, going from a smaller force to a higher force. So what they found out that at a lower force of 5 piconewton, as we can see here, we see a straight line. That means the CRISPR-Cas9 is binding to only one location. But as the force was increased, the like as we saw in the video before, the chymograph verifies that there are multiple offsite binding of the CRISPR-Cas9 on the DNA molecule. And this entire process is reversible, which means that when the DNA was relaxed from a tense state to a relaxed state, again the CRISPR-Cas9 bound to only one location. This is a more quantitative representation of this entire chymograph, which shows that the offsite binding of Cas9 when the DNA under tension is pretty significant. Moving on, I will talk about another application where C-trap has been extensively used in the past few years, is biomolecular condensates. So biomolecular condensates are membraneless particle droplet-like structure which exists inside the cell, and it is kind of postulated and believed that these droplets could be responsible, or might have biological implication. For example, they are kind of highly studied, uh, postulating that they could have, they might be related to DNA packaging or DNA compaction. So what they did in this experiment here, which they found out, is they wanted to study how this biomolecular condensate here, it's a fused sarcoma protein, which is so it's a complex of fused sarcoma protein and RNA molecule, and they wanted to study how these biomolecular droplets interact with each other in the presence and absence of a molecular crowder, which is PEG, what they used here. What they found out that when they trap these two droplets and bring them closer to each other using the optical trap, they would easily fuse in the absence of the molecular crowder. That means when there was no PEG there. But when they put in PEG in the solution, what they found out that the affinity of these two droplets to become affinity of these two droplets to fuse onto each other was kind of diminished in the in the presence of a PEG linker. Another area of study where C-trap is extensively used is to study protein folding and unfolding experiment. Precisely saying that to study the dynamics of protein. So protein folding has been the holy grail for the biophysicists for multiple years, and computational biologists and scientists from different disciplines, they wanted to study how a protein folds, or how the dynamics of a protein structure evolve over time, because it has been related to multiple biological implications, because every folded protein forms a particular function. So it could have multiple biological implications, for example, in drug design. So the way a protein folding assay, which is done in a C-trap, is there are two beads which are taken, and the protein molecule is tethered between these two beads using a stiff DNA handle, and then the beads are pulled away from each other, which generates a forced extension curve, as we can see here. This is the worm-like chain behavior, and at this point the protein unfolds, and then when it is relaxed, the protein folds back again. From this data, we can also using the C-trap, we can also measure how the force on the protein changes over a period of time. And correlating both of these two, we can determine what is the time up until which the protein remains in the folded state, and what is the time which is required to unfold the protein. So which gives us the k on and k off, precisely speaking. So once we know the k on and k off, we can estimate the energy landscape which is associated with the like with this protein. That can help us, in multiple kind of to address multiple questions related to this particular protein. For example, here I will share with you a data which was published in Nature, I think, where they studied the ADK kinase enzyme. So one of the, as we all know, that the enzyme has open and a closed state. So when it opens, it binds to the substrate and then it closes. So until this experiment was done, which is a combination of a C-trap and a molecular dynamic assay using computational method, what they found out that this ADK kinase undergoes conformational changes between the open state and a closed state. Uh, so it goes a conformational change of 17 angstrom, which is related to its function, which was not determined using the X-ray crystal structure data which was available before. What the X-ray crystal structure data showed was only the closed state. So they postulated from this experiment that this ADK kinase enzyme undergoes unfolding from this closed state to a open state, which facilitates the binding of this particular enzyme to a particular substrate. Another area where C-trap is extensively used is to study the interaction of motor protein with biological filaments, for example, microtubule or actin filament. The reason why the interaction of microtubule or with biological filaments are studied a lot nowadays is because of its association with different type of neural diseases. So to do this type of experiment, one can functionalize a kinesin molecule, which is shown as in the cartoon representation of the kinesin is shown on the left here. It is bound to a bead, and then once this bead is trapped by the optical trap, this bead is brought close to the microtubule. And once this bead is brought close to the microtubule, the kinesin molecule starts to interact with the microtubule, which can be seen using this video here. So this is a IRM image, which means interference reflection microscopy. This is a label-free technique which does not require any kind of a dye, and this can be done. This can be combined with the optical trap and the C-trap. And as we can see here, the fluorescent bead is nicely working on the microtubule. What we can find out from this experiment is the load and the velocity with which the microtubule is walking, or the kinesin is walking over the microtubule. We can also measure the interaction forces between the legs of kinesin and the microtubule, which could have implication uh, and could address questions which are related to kinesin from a disease sample or a normal sample. And finally, I will just share this slide with you because I'm not going to talk too much about it because Yen is going to talk more about the mechanobiology part. So, as we know, cell moves. So when cell moves, it might anchor along its way like other components in the microenvironment, and this anchoring depends upon the elasticity or the stiffness of this particular cell. It also interacts along its way with other member of the microenvironment, for example, other cells or collagen, and they have been related to the biological implication. For example, it is postulated that in case of the cancer cells undergo change in stiffness compared to the normal cell. And it is also hypothesized that the cancer cells bind more strongly to the member of the microenvironment. So on the left hand side here is a cell which was fluorescently labeled, and we study that there was a one trap bead was brought in the vicinity of this cell, And as this cell proliferated, it interacted with this bead, and from this the forces of interaction of the filopodia-like structure from the um, cell can be measured. It also gives us a idea about the stiffness of the cell as well. One can also measure the interaction of a particular ligand with respect to the receptor which are found on the cell surface. In this experiment, what the user did is that they functionalized this cell with biotin and the surface of the cell with streptavidin. And and once they brought the cell closer to the cell, which is we cannot see the bead here because it is not fluorescently labeled. The biotin and the streptavidin interaction takes place, and once the bead is pulled away, we can see the component which are extracellular and intracellular also move along its way towards the direction where the bead was moved. Here, um the proteins which are on close to the cytoskeleton were labeled with a blue dye, while the protein which were beneath the membrane were labeled as red. So one can also use a C-trap to probe biological cell, and also using fluorescence they can understand the like what happens to the biochemical or biostructural property of the cell when mechanical force is applied. Finally, although as I said that C-trap is extremely easy to use, but we do understand that the user has to prepare sample and perform the data analysis. So to reduce the barrier of sample preparation, Lumicks has a biochemistry kits and service store, which through which a user can obtain different type of DNA and protein sample we already discussed, like biotin related and streptavidin coated DNA and lambda DNA, which had biotin in it. So a lot of the sample work barrier can be reduced with the help of the Lumix biochemistry kit and service store. The second part is the data analysis part. So we always work with the user to perform the data analysis. Also, there is a site called Harbor site on Lumix, where a user can have access to different type of scripts which they can modify. They can make their own script to perform a particular type of experiment. Also, they can. There are a lot of data analysis scripts available as well, which a user can use to uh, design automated data analysis and custom built data analysis procedure as uh, scripts as well. And finally, this is my last slide, and I would like to share it. And it kind of makes me really excited to share this. That till date, there are, I think there are, we have already crossed the 100 publication using the with using the C-trap or the DSM technology, and the average impact factor of most of all these journals are more than 7, which means that over a 4-year period, every 6 months we have been publishing close to 25 journals, very high impact factor journals, which gives an indication that how remarkable this technique is, and also gives an insight to us that where the field is heading towards. That we are more and more interested in knowing the like how a biology like what are the mechanics or how related to a biological system or a biological function. With that, I will pass it to Yen, who is going uh, to tell us more about the Cell Avidity uh, instrument. Thank you very much.
Yeah, thank you. Let me set up my hold on one second. So, I mean, Samrat, I think you need to stop sharing so I can share.
Okay, sure. I will do that. Stop sharing. Yep.
Great. All right. Thank you.
Perfect.
Okay. So then, yeah, let's let's move on. And yeah, thanks everyone for attending. My name is Jens Eberlein, and I'm the technical lead for the Cell Avidity part here at Lumicks. And I would like to zoom out from the molecule level and shift the focus on to a different topic that is vital to the success of cell-based immunotherapies. And that is really how we can measure engagement between an effector cell and a tumor cell. And by quantifying that engagement or strength of binding, also called cell avidity, we'll explore what it can tell us about the future function of these cells. Yeah? So we'll discuss our current understanding of how affinity and avidity relate to function and how we can use acoustic force to quantify this interaction. Okay, so I would like to start with a question, which is, do we have the right tools and the best information to select and develop the best immunotherapies? Yeah. And I'm asking this question because, unfortunately, most CAR-T therapy trials fail for a number of reasons. And the big question is, why does this happen? And one of the major reasons really is that our current in vitro assays fail to accurately predict in vivo assays. And as an example here, I'm showing some data from an article that I'll come back to later. And so here we'll focus on two different CARs that kill equally well in vitro. And one of them secretes significantly more cytokines, humankines, and cytokines here. And so there's a disconnect, right? And surprisingly, what you can see here, when you're going in vivo into an in vivo mouse model, you can see that one of the two is a clear winner, right? So it doubles the survival of the mice. And so the question really is, why is that? And so in essence, the traditional biophysical methods primarily focus on affinity, as shown here on the left-hand side. You're probably very familiar with the term. And what we are doing here is essentially looking at single antigen and antigen receptor or single ligand receptor interactions. Yeah? So it doesn't have to be in the immune context, but and this is also done in a forceless environment. So most often the gold standard is a surface plasma resonance, or SPR, and so you're essentially looking at on-off rates in a forceless environment. Yeah, and you can calculate the KD here, and this can be extremely useful in the biopharma to screen large panels of potential binders. Yeah. And however, what is really missing is a metric we call here overall cell avidity, so the ability to measure the overall strength of interactions between a diversity of receptor ligand pairs on the cell surface. Yeah? So as you can see here, for example, you have a engineered CAR-T cell in blue that is interacting with a cancer cell, and it's not just the engineered CAR receptor here that interacts with your cancer cell, but you have numerous other co-receptors, LFA1, ICON1 interactions, et cetera, that are contributing to the overall cell avidity. And really, this cell binding strength determines the initiation of what we call the immune synapse, and which is important for all downstream events of T cell activation. And again, I don't need to too much over this, but while the effector cell is engaging with its target cell, right, we have numerous processes going on in sequence and also in parallel. And what is really important here is that the strength or weakness of the initiating binding event between your effectors and targets really amplifies the signal and determines what happens downstream. And Samrat already mentioned that, yeah, there's some forces involved at the cell level as well in his closing slide. And this is, yeah, really a central theme here in immunology, right? So we have, for example, cell receptors that can have similar affinities as measured by SPR. But if you are applying a pulling force on these, you will see that there are some that have that are slipping away once force is applied, they're forming so-called slip bonds. And then you have some TCRs that are forming these Chinese finger lock principle interactions, these catch bonds. Yeah? So when you're applying a force, actually it enforces the interaction. Another example where forces play a big role in cell-cell interaction is also once the T cell docked onto its target and is ready to recognize the target and is ready to kill, it pushes and pulls on this interaction, and this has been shown to be important for depositing granzyme perforin into the cell membrane of the target. So there's a better perforin integration, which then leads to better deposit and higher deposit of granzyme B, and therefore better killing. Yeah? We encounter forces all over the place in immunology. And up until now, we really did not have a good way of measuring these cell-cell interactions, looking at overall avidity. And so this is where we come here into play, Lumicks as a company. So we developed a Cell Avidity Analyzer called the z-Movi. And what it allows us is to exactly look at the cell-cell interactions, and most precisely how much force it takes to pull two interacting cells apart. Yeah. And so how do we do this? So here's a schematic of our instrument. On the left-hand side is a picture of our instrument. And at the core is really, at heart is our lab on a chip. That's shown here. And then also shown here in the side view. And switch real quick to a different pointer here. So the instrument itself is fairly small. There's a very small footprint. It's about the size of 2 toasters. So it's easily fits on your lab bench. And the instrument itself has a microscope in it, and we're looking at bright field, and we're employing a fluorescent far red tracking dye to look at our effectors. And you can think of, if you're familiar with killing assays, you can think of our assay as setting up a killing assay. So where you are plating in our microfluidics chip a cell layer of target cells, like a single sheet, and we call it a monolayer, as shown here on the bottom. And then we are flowing in our fluorescently labeled effector cells. And these are the ones that we are tracking. Let them briefly interact, so in anywhere between 2.5 and 10 minutes, let's say. And then we are applying an acoustic force ramp. So we're employing this piezo element that you can see on top, bonded on top here in orange. And when we're flowing electric current through it, it creates standing waves. And these waves transduce into the chamber and creates an upward pulling motion on your cell. And since your target cell, so these can be adherent or non-adherent cancer cell lines, since we are making them adhere firmly to the bottom, the only cells that can lift off are your effector cells. And so after a short co-incubation period of 5 minutes, we are applying an acoustic force ramp where we are ramping up the pulling force from 0 piconewton to 1,000 piconewton. So in about 2.5 minutes. And during that time, we are basically tracking each individual cell and look at what point does it detach from its target. And when it detaches, it migrates up into this acoustic node shown here. So it's the 0 force field where they're essentially just hovering mid-chip, if you're looking sideways here. Yeah, so that it's floating in the media here and accumulating there. And the chip itself is reusable, so we can do 125 runs on this chip. And also, what is really interesting is that we can also reuse the same target cell layer that you see here sequentially. So on the same chip here, let's say you're looking at 5 different CAR T cells that you want to interrogate the binding of. You can just flow them on sequentially here. put them on, interact, and then do a force ramp and look at the ability it took, the force it took to overcome the binding. So we move on. So this is a top-down view of our field of view that the instrument is analyzing. And we aim for about 400 cells in the field of view, 400 effector cells that we are tracking. On the left-hand side here is a negative control, so non-tumor targeting T cells. And when we are applying the force, you can see how the cells are being lifted off from their original position and accumulate in these acoustic nodes. Okay, for a high avidity sample, so targeting cells, and you can see here on the right-hand side, cells are being lifted off, but a lot of them are still at the end of the force ramp, stuck to remain bound to their target. So let's take a look at the workflow again. So typically what you do is in the morning, you start at 9 o'clock, you're seeding your target cells into your chambers and let them adhere for 2 to 3 hours. You just stick it back into your normal incubator to let them rest. And then in the meantime, you're simply labeling your effector cells. And then in the afternoon, when you're ready to do your experiments, you just load your chip onto the z-Movi and then flow on your first T cells that you, for example, that you're interested. We can also work with NK cells, CAR NKs, cell engagers, TCR transgenics, and so forth. So the system is very flexible. So you flush on your T cells, let them incubate, interact, then you do your force ramp, and then you're ready to float the force ramp to just flush out any unbound CAR T cells, for example, and then you're ready to repeat the process with the next CAR. And this way you can quickly screen 4 to 6 different CARs on the same chip, and then you are just moving to the next chip that you pull out of your incubator and start the process over again. So let's look at how the data, the output actually looks like. So the instrument generates a file that's very similar to a flow cytometry file for those of you who are using flow cytometry, so very similar to an FDS file, where you have the X and Y coordinate for your cell, for your effector cell, and then on the next column over, you have the force of detachment. And so it's a simple CSV file, and we can use that to compute different graphs. And so the most commonly used one is shown here on the left-hand side, where you have on the y-axis the percent bound T cells over the force. And so we always want to have ideally a negative control, either an untransduced sample that is not recognizing your target cell line, or have a target cell line that doesn't express the antigen. And so in black is the typical curve of a negative untransduced cell line here, untransduced effector. And as the avidity increases, you can see it's shifting up, right? So a low avidity sample here in green, shown here in green, and the high avidity in red. You can slice through the data, as shown on the dotted line, either where the untransduced levels off, or at the max force at the end of the run, and then create bar graphs. Again, this is all single cell data. So if you're looking on the right-hand side, you can also plot this in violin plots. Okay. So just wanted to give a little background on the technology itself before moving on and showing you some actual data that was generated with our technology. So as we discussed, in vitro killing and cytokine secretion assays are often unable to discriminate between different immunotherapy designs, and there's an urgent need for new methods, right? And the initial cell-cell interaction strength between an effector cell and its target informs all downstream events, right? Immune synapse formation, the quality thereof is very important. And our platform allows you to rapidly measure the cell avidity at the single cell level for about 400 cells in under 10 minutes. So now let's move on and look at some practical examples here of how our tool has been, our assay has been used. And again, as I mentioned, we can not only look at cells, but also at TCR transgenic cell lines, CAR NKs, and cell engagers, and many more. I want to start off with an example here out of the TCR field. This was done by Natalie Rufer from the University of Lausanne, where she was interested in engineering and NY-ESO specific TCR. So she had a wild type TCR shown here on the left-hand side. She did affinity tuning. So she increased the affinity as measured by SPR of this TCR and created higher and higher affinity versions. And when she was plotting the affinity as measured by SPR over T cell function, this could be interferon gamma secretion or killing or IL-2 secretion, she saw that it's a bell-shaped curve. So you have, as your affinity maturing your TCR, you have a gain of function up to a certain point. And when the affinity itself of the TCR is too high, you see at some point a drop. So you have this bell-shaped drop. There's a non-linear relationship between affinity and function. So we went to look if there's a better correlation between avidity and function here. And again, these are our binding curves that I've showed earlier. So where you have the 1% of T cells here on the y-axis over the relative force. We're starting here with a low avidity example, the wild type TCR. So you have some separation between the negative control in black and the peptide pulse, the positive cell line that the TCR can recognize. So these are peptide pulse and NAH cells. Intermediate version of the TCR is shown here in the middle, where you can see now there's a shift, right? So they increase the affinity of the TCR, which translated in this case into an avidity increase of the TCR interacting with the target. So there's a nice separation between unpulsed and peptide pulsed NAH cells here. Now, what's interesting is looking at a very high affinity TCR, you see also the avidity increase is nice compared to the wild type TCR. However, there's also an increase in the background binding. Now you see the black line here moving up as well. And so the signal to noise, so the difference between those two is narrowed for the high affinity TCRs. So when we are now using our avidity and dividing it pulsed by the unpulsed, you can quickly see here that the optimal maturity, the TCR, is the one that has the best signal to noise ratio, and we call this an avidity score here. Long story short, if we're looking at TCR affinity over our avidity score, again, you see a bell-shaped curve. So there's a nonlinear relationship between avidity and affinity. And looking now at avidity, at our avidity score over T cell activation or killing efficiency, you see a more linear relationship here. So avidity is a better, correlates better with function than affinity. Now I want to move on to some T cell examples here. So chimeric antigen receptor transduced T cells. And this work was done by Marcella Mauser's lab at MGH and was recently published earlier this year. And so she was interested in modifying her CD27-based CAR T cell, which is prone to proteolytic cleavage, to shedding of the receptor in the tumor environment, because it has a putative cleavage site here, or cleavage site, yeah. And so their project was to, the scope of this project was rationally engineer a CAR construct that is not being fed. And what you can see here is, so they started with the original CD27-based CAR, then created a truncated version to lube out the cleavage site, then designed a deleted version where they don't have a stark region, introduced a flexible linker, and then swapped out the hinged transmembrane domain with the CD8 hinged transmembrane domain. So they went on to look at various assays, and one of them is shown here, where they did a serial stimulation assay over 28 days, followed by a killing assay. And as you can see, after all this work, essentially all of the constructs are lying on top of each other. So the assay cannot differentiate between the different CAR designs, with the exception for the truncated version here, which performed poorly in killing. On the contrary, let me see. Yeah, on the contrary, if we're running our assay and look at the relative avidity to native, so meaning that here at 1.0 is the avidity curve of native divided by native, gives you a 1.0, and everything that is above 1.0 means that the avidity itself has increased compared to the original native car. And anything below 1.0 means that the avidity was actually decreased by engineering, by the engineering efforts. And as you can easily see here, only one of the CAR-Ts, the CD8 hinge and transmembrane domain version, improved the overall cell avidity. So they went ahead and studied, went into in vivo. And here also, long story short, what we could show is that the only one that improved survival of the mice was the CD8 hinge and transmembrane domain. It's shown here on the right-hand side, the red line here. Yeah. So next they went ahead and looked, okay, so how does the in vivo data at day 14, where they still have all the groups, the mice alive, how does the that tumor burden correlate with traditional assays, in vitro assays? So they compared the correlation between the day 14 data of the in vivo data, and an R-square of 0.6, looked at the interferon gamma secretion, R-square of 0.55, and then compared it to our assay to cell avidity readouts and got an R-square of 0.9. So we were the most predictive of in vivo in this case, in this study. Yeah. Another example here, also from Marcelo Moss's lab, was one of the first early adopters here in the US, is around avidity escape. So this is work done in her lab and published in Nature earlier this year, I think in April. She was interested in tumor escape mechanisms of solid tumors. So one of the issues is, most of you might know, is that it's somewhat easier to design CAR-T therapies for liquid tumors, but solid tumors, we haven't gone that far yet. So there's a lot still to be done. And so she wanted to see, okay, if we're using a genome-wide CRISPR screen on the tumor side, can we identify any receptors, any factors that confer resistance to CAR-T-mediated killing in solid tumors? And so, long story short again, so when they identified interferon gamma receptor 1, so if interferon gamma receptor 1 is knocked out in solid tumors, and they did this for several different tumor cell lines, the CAR-T cells can kill. They went ahead and used our assay and looked at relative avidity here, shown here, and could show that compared to a wild-type tumor here in black, the interferon gamma receptor 1 knockouts had reduced cellular avidity, yeah? And they also can kill. So now they went ahead and restored cell avidity by doing a pathway analysis and identifying ICAM1 as being downregulated in the absence of interferon gamma receptor 1. And so they reintroduced it, overexpressed ICAM1 in the knockout mice, in the knockout cell line. And what you can see here, the black line is wild-type cell line, and then the red ones are the interferon gamma receptor 1 knockouts, rescued with ICAM1 overexpression to restore ICAM1 overexpression. And this then led to a restored killing. So now, shown here in cell volume, interferon gamma receptor 1 knockouts in dark red here, they can't be, the cell volume stays the same, they can't be killed. And now overexpressing ICAM1 in the interferon gamma knockout gives you a phenotype essentially the same as the wild type. So now they can be killed again. Yeah. Okay, so coming to the very last examples here. This is I like to always show this to show the flexibility of our assay. So in the beginning, I mentioned we typically do short co-incubation periods. So it's just enough for them to interact, the effect on target, and then we pull on the interaction and quantify this. There are other questions you can ask too with our assay, and this one here is an example of binding kinetics. So there are two car designs that have the same surface receptor, the same SCFV, but they differ in the endodomains. And so here we're looking at co-incubation profile from 2.5 minutes to 10 minutes, 20 minutes, and 40 minutes before applying the acoustic force ramp. And what you can see, intuitively, you would think, okay, they have the same surface, right? Molecules on the surface, same SCFV that recognizes the antigen and attaches, they should have the same avidity. But what is really interesting is that you can see different kinetics here. So, for example, the green endodomain B here is a faster on, so higher avidity, higher binding. Early on, it peaks at 20 minutes and then drops off. With the A, you can see a little bit of a delayed overall increase in avidity, also peaking at 20 minutes and then being more sustained. What is, I think, very interesting here is that you not only have outside-in signaling that's going on, but inside out as well, once the two cells are engaging. Yeah? And you have your synapse formation, and the quality of the synapse also informs the avidity here. And I'm closing with a real-world example here. So this was a study, this was constructed CAR-T construct here from community, which they had to pull from the phase 1 trials because of adverse conditions. 2 patients died. So they went ahead and did engineering of that CAR by changing out the costimulatory domain. So the same, again, surface CFV domain, but then swapping out the costimulatory domain led to a reduction here in binding and overall avidity. And hopefully this will also reduce the CRS, the adverse side effects. Yeah? So again, here by manipulating the intracellular domain, you can also manipulate cell avidity itself. Okay. So just a quick summary here. Cell avidity, I hope can convince you here, is the new biomarker to provide unique and mechanistic insights into immunotherapy design. So we've looked at a TCR-based example where avidity correlated with the effector function. T, we are looking at also correlation was better compared to conventional assays like interferon gamma and killing, and was the most predictive of in vivo function. And also, cell avidity measurements were used to provide novel insights into tumor escape mechanisms and led to the term avidity escape. Okay, and so there is a need really, and I haven't had time to show more data on this, but for tuning cell therapies to really the desired avidity level for ideal function and persistence. And our instrument, as you may know, is the only platform out on the market that is able to measure cell avidity and allows you to apply this for a range of experimental questions. Okay, so with that, I will conclude our presentation, and thank you everybody for listening.
Thank you. Awesome presentation. I always love when you get such beautiful images while still getting really awesome data. It's nice when it goes together. But yeah, so if anybody has any questions, feel free to drop them in the chat, the Q&A. We'll be answering them now.
Okay, and while we're waiting for the questions, if there are any, I wanted to say that if you would like a copy of the presentation, please email either me, you can see my email here on the screen, or the email that was on the last slide, and we'll gladly send you a PDF version of the slide deck. There you go. Okay, so no questions. However, if you have any questions afterwards, and Alex, can you confirm that this presentation will be available online?
Yes.
Right? So yeah, feel free to rewatch it, share it with friends, colleagues, and yeah, please get back to us if you have any questions and interests, or you would like to discuss your specific applications, either for the C-Trap or for the z-Movi technology. So no questions. We are over 5 minutes over time. I think it's time to thank everyone for taking time to be with us on this pre-Thanksgiving day. I know everyone is focusing on the family and friends and the day tomorrow. So with that, I would want to maybe thank you, Alex, for giving us this opportunity to present to Rutgers community. Thank you so much.
Yeah, of course, and thank you for giving a great presentation. This was very informative, and hopefully we'll have a lot of people reaching out to you in the follow-up.
Thank you. I hope so too. And yeah, and the last thing I would say, I want to wish everyone happy Thanksgiving and hope you have a fun, cozy, and relaxing Thanksgiving with your friends and family. So thank you, and
All right.
hope to hear from you soon.
All right, and thank you.
Bye. Thank you.
Hey, have a great day.
Thank you.